some of us, either ourselves or our family member or our patients, might have very mild or moderate glaucoma. It's actually the leading cause of irreversible blindness in the world. It's a major cause in this country, in the developed world, but it's the absolute leading cause of irreversible vision loss in the world. And the problem is that so many people who have glaucoma don't even know they have glaucoma. And so this becomes a major, major issue. How do we diagnose glaucoma? And for the people who have glaucoma, how do we know if they're getting worse? How do we know if they're about to get worse? Now, there are some things that give us hints, like risk factors. And these can be things like increasing age and increasing eye pressure. Those are going to be the major two risk factors. But of course, we can't just look at a patient and say, well, you're a little bit older and your pressure's a little bit higher, so you must have glaucoma. They're just risk factors. We have to really diagnose it based on other features. These factors are not sufficient for diagnosis or for monitoring progression. What's damaged in glaucoma, that's the part that we have to follow. The vision loss in glaucoma is due to the dysfunction and eventually the death of cells in the retina called retinal ganglion cells. And they're axons, they're fibers that connect the eye to the brain and carry all of the visual information through the optic nerve. That's what degenerates in glaucoma. And fundamentally, at some level, that's what we're trying to measure. So how do we measure those today? How do we distinguish the dysfunction of these cells, the dysfunction of the optic nerve, from the eventual death? These are, it turns out, very hard questions to answer. So what are we doing today? Today, for function, we use the visual field test. And of course, many of you have suffered through this exam. It's very hard to do. It's very subjective. As we all know, if you haven't had your coffee that morning, you're not going to look as good on the visual field. And then your doctor's going to say you're getting worse, and you're going to say, no, I just forgot my coffee. And so it spits out complex data like this, and this is an example of an eye that looks pretty good, an example with mild glaucoma, and the more black you've got, the worse things are getting. But we ask our patients to take these visual field tests every year, maybe twice a year, rarely much more than twice a year. And as we said, it's a very subjective test to be able to tell how you're doing. Also, it only tells us whether you have glaucoma or whether you're getting worse after it's happened. After the damage is there, then we can detect it. So that's not that fulfilling for patients who want to say, wait a sec, tell me I've got damage. Tell me when I'm going to get damage, not after I've already had it. We can do these predictive progression analyses. Like if someone's declining, we could say, well, hopefully, they're probably going to decline at about the same rate. So here's what the next 5 years will hold. But these are very kind of messy predictors. They're not very good markers for the disease. So then we look at the structure of the optic nerve. That was function, this is structure. When we look inside your eye, this is what we're looking at. This is the head of the optic nerve. Here's a little bit of healthy tissue left in this patient's eye, but a lot of missing tissue that has already degenerated in this patient with glaucoma. We use other imaging analyses. This one's called an OCT, or your retinal nerve fiber layer. It measures the thickness of the fiber layer that's going into the optic nerve. And again, it's a very gross measure, and it tells us about damage after it's happened. It's good because it's a little bit earlier than the visual field damage on average, but it's still catching things late. So the take-home message is that everything we have in the clinic today, it's not enough. It's not really capturing what we need. So what do we really need? We want to be able to diagnose patients with a reduced clinical expertise. This shouldn't take the best glaucoma specialist in each major city to figure out if this patient has glaucoma. There are simply too many patients. So we need something that's fairly easy to use and can really spit out an answer. This person has glaucoma. We need something for treatment that has nothing to do with intraocular pressure. We need cures for this disease, and we're going to come back to that point. We want to detect this disease earlier, and we want to know who's getting worse before they've gotten a whole lot worse. We want to know in shorter intervals. And we want to even detect the risk of progression. We want the patient sitting in front of us, we want to be able to say to this patient, the next year, it's going to be a bad year for you if we don't do something today. We don't want to be in a situation where we say, this past year's been a bad year for you. We want to say, we're ahead of the game. Next year could be bad, but we're going to intervene today. So all of that gets served by what we call biomarkers. And biomarkers are just ways to measure the disease, the risk, what's happening to those retinal ganglion cells, what's happening to their axons in the optic nerve. We want to diagnose before the vision loss takes place. We want to detect very subtle progression so that we can adjust treatment faster and in a patient-specific manner. And here's an important point I want to bring home. We need better biomarkers to develop new treatments or cures. And I'm going to give you an example of that right now. The problem is that it's very hard to test for treatments or cures, things that we call like neuroprotection in glaucoma. And the reason is that on average, it's a slow disease. It can take a long time to measure, and it's hard to make clinical trials for new cures if you're going to tell the company making the new cure, oh, and by the way, it's going to take 4 years to do this clinical study. You know, they don't want to hear that. They want to hear about short studies. And frankly, patients want to hear about shorter studies too. They want to know quickly, hey, is this helping me? But if we could detect dysfunction and find cures that reverse this dysfunction, then maybe we could detect that on a very short time scale, and this would allow us to figure out cures faster. So I'm going to give you a very short example of how a better biomarker is going to speed the cure. Here is one particular drug called ciliary neurotrophic factor, or CNTF. And we know from animal studies that this promotes optic nerve regeneration. This can regenerate optic nerves in animals all the way from mice to monkeys. A phase 1 trial in 11 patients with glaucoma was performed where a little implant that spits out CNTF was put into patients' eyes. And this spit out CNTF, well, it's still doing it in those patients' eyes, but it was studied in these patients for 18 months. And we studied structure, we studied function, we studied everything we could think of. And here's one of the upshot slides. When we looked at the visual field index on those visual fields, the implanted eye seemed to get a smidge better over 18 months. That's pretty good, considering that in glaucoma, normally nothing gets better. Whereas the unimplanted fellow eye got a smidge worse over the same time period. But it was very hard to detect because, again, these visual fields are very variable, and so it could be very challenging to figure this out. If we had a better biomarker, we would know very quickly, maybe even in the first few months, if the CNTF or another drug that needs to be tested worked in these patients. And if we had that biomarker, more companies would say, hey, I've got a product, and if it's a short time to test, let me test it. So we need to measure improvement or decline faster and better. So what are the steps the Glaucoma Research Foundation has taken? Number 1, assemble a team. And Monica was kind enough to introduce the 4 of us, and you're going to hear a little bit about progress that the group is making. Andy Huberman and I, Jeffrey Goldberg, were at UC San Diego, Alf Dubra at Medical College of Wisconsin, and Vivek Srinivasan at UC Davis. So they're coincidentally a little bit of a tilt towards the left side of the country, but it's just a coincidence. Okay, so step 2, we've got number 1, the team. Step 2, identify new biomarkers. And the premise of this team is to bring biologists together with optical imaging engineers, physicists, people with that kind of background, draw from the biology of glaucoma and visualize with engineering and optical engineering. So in the first 3 years, actually, the team has made a number of what we think are really exciting new discoveries.
My name is Andy Huberman. I'm at UC San Diego, where I'm a professor of neurosciences and biology, and at the Salk Institute for Biological Studies. I just want to tell you a little bit about some of the progress related to the biomarker initiative. My goal here is really to highlight 2 things. One, what we think is a very exciting discovery that we're going to leverage toward attempts for better clinical treatments and better assays within the clinic for detecting glaucoma, for all the reasons that Jeff mentioned. And the other one, which I think is a strong underpinning of everything that you're hearing today, is the unique nature of this collaborative effort. You know, just without getting into the details about how research is typically done, all I can tell you is that it's quite rare for a team of both biologists, engineers, imagers, and a clinician to work together so closely on a common goal. And that's a very unique thing because it's just simply not the way that the federal granting structure has been organized. And so what's really been wonderful, I can tell you now, having worked with these guys for the last 3 years and with the Glaucoma Research Foundation, is that all 4 of us and the many members of our laboratories, who of course are the people that are doing all the hands-on work, are aligned towards a singular goal. And that's extremely unique. And I'll just mention as another side, and then I'll get to the data, is that this year, the National Eye Institute, which is paid for by your tax dollars, has identified audacious goals, an audacious goals initiative. One of the major audacious goals, and there are really only 2, is to identify better ways to treat and understand and cure ganglion cell loss in diseases like glaucoma and other optic neuropathies. And they're already asking us, this group, and I've had some long conversations with the directors of the Audacious Goals Initiative, how to assemble teams like this. And so they're turning to our group to understand how to take scientists and allow them to work together. And I think the rapid progress that we've made is one of the reasons why they're accessing us for that information. So we're hoping that this model will ripple out to many, many groups. Okay, so now into the data. What you're looking at here is one retinal ganglion cell, an example of a retinal ganglion cell. So what you see in green is a retinal ganglion cell. These are absolutely beautiful cells. To give you a sense of the scale and the context here, your retina, which sits in the back of your eye, is what informs your brain about everything in the content of the visual world. Put differently, your brain knows nothing about the visual world except what the cells in the retina tell it. And that's an amazing thing. This tissue, the retinal tissue, is about as thin as a credit card. And it alone informs you about everything that's going on in your world. Without it, and without the retinal ganglion cells which connect the retina to the brain, as Jeff mentioned, no vision occurs. And so this retinal ganglion cell is a very tiny cell, and there are many others like it, which are labeled here in blue. This one happens to be labeled in green. So my laboratory, and for many years now, almost a decade now, I've been working on tools to try and label these cells more specifically so that you can see them in all their entirety here. And they're very beautiful cells, if you ask me. Why would we want to label these cells? Well, it turns out that by nature's design, they're clear so that light can pass through to the photoreceptors on the back of the eye. So here's the fantasy. Instead of having to do visual fields or other indirect measures, the fantasy would be for a patient to go into the clinic, and based on methods and tools that we've developed, biomarkers, for that patient to sit down and we could say, okay, Mr. or Mrs. Jones, you have 100 ganglion cells in this little patch of retina, but last year at this time you had 150, and we need to treat you. Or you had 100 last year and you still have 100 now, and so the treatments are working. So these are the sorts of sensitive measures that are not available yet, and we need to develop tools like the ones that we have in animal studies to visualize the number and type, and I'll mention why, type of ganglion cells that are present in your retina and my retina. That would be a wonderful thing to have. Why? Well, first of all, we would know whether or not you were losing ganglion cells. Second of all, what I haven't told you is that there are many different kinds of ganglion cells. There are about 1 million of them in each of your eye, and they're absolutely essential for vision. But there are about 20 different kinds, and each one looks very different. Some are very large, some are very small. And one idea that's been kicked around in the glaucoma, both clinical and research literature for a long time now, is this question: are there specific types of ganglion cells, maybe the big ones, maybe the small ones, that are being lost at early stages of the disease? If that were the case, and this has been argued back and forth for many decades, and if there was a way to detect the loss of specific kinds of ganglion cells, let's just say the big ganglion cells, that would be a wonderful thing, because you could imagine, because the different types of ganglion cells respond to different things in the visual world, some like dark objects, some like moving objects, some like bright objects, you can imagine a visual field test that was very sensitive for detecting ganglion cell loss long before you had holes in your visual field. That would be a wonderful thing to have. And thanks to the hard work of a postdoc in my lab, Rana Eldanoff, I'll just tell you about this one very exciting discovery that Rana's made. There are many different kinds of ganglion cells. I'm showing you 3 different types here to just give you a sense that here's a very big cell. It's red because the connections that it makes sit at a very particular location. Think about it as a particular floor in a building within the retina. A different kind of cell is much smaller. These are normal, healthy cells, and this one looks very different than the other 2. So in glaucoma, are specific types of cells being lost or changing? And it turns out there are. So what's really impressive, what Rana's discovered, is that there's one type of cell, like this one, which is not impacted by elevated intraocular pressure. These cells are refractory to intraocular pressure. It looks very much like the normal cell. But the other types of cells, which we call off cells, are changing very early on in the disease. Why is this interesting? Why is this useful? Well, what we're imagining are visual field tests that specifically probe the function of these off cells. In other words, visual field tests that rely on dimming of lights. And we're now in the process of talking to people who do clinical psychophysics to try and design such visual field tests. In any event, we think this is an important discovery and in many ways resolves the issue of whether or not there are specific kinds of ganglion cells being lost early in the disease. And the answer is yes. I'm going to now turn over the stage to Vivek, who's going to tell you about some other impressive biological discoveries by the team.
And so this finding that Andy had has led to a number of new ideas in the team about how we can get at visualizing this layer, either visualizing the cells directly or indirect measurements of what's happening in this off layer. And as it turns out, this off layer is also a site rich with blood vessels and has had much higher energy usage than the other layers in the retina. I'm going to talk a little bit about the imaging approaches or the new approaches to detecting glaucoma that this has led to. But first, I'd like to say we are developing new visual field strategies. So the idea is think of it as like a visual field 2.0 that's going to selectively measure the off pathway. Other approaches are to develop imaging methods. So imaging methods means just a fancy way of saying take high-resolution photographs of the back of the eye. And so one idea is to develop ways to look at blood vessels and how they respond to glaucoma. Another is to measure retinal ganglion cell metabolism, energy use, and their connections, and how those respond to disease. And so here's an example of vascular imaging in my lab where we've developed a way to graph the three-dimensional vasculature in the rat. And so we can measure the vessels or the blood vessels very precisely, supplying the layer where Andy saw those early changes. And so we've been working on developing methods that we can use to isolate those vessels in those layers and study how they change in glaucoma. And if, in fact, the blood vessels in the off layer change early in glaucoma, just like the off sublamina does. And so there's been a huge push from the board to study human imaging or to move our imaging techniques into humans to help patients faster, and I think rightfully so. So I'd like to talk about a few imaging approaches we're developing for translation. One is this idea that we can use an FDA-approved compound. So FDA is the Food and Drug Administration, which has to approve any devices or therapeutics. And the idea is we can use an FDA-approved compound that's already been approved for a different purpose and use it as a contrast agent to improve our imaging. So one idea we've looked at here is we're using a dynamic contrast-based OCT imaging with intralipid as the contrast agent. So intralipid is already FDA approved for nutritional uses for intravenous injection. And the idea here is we can use the extra signal that contrast agent gives us to improve our imaging of blood vessels. And actually here we're able to actually now measure blood flow in individual retinal layers with high resolution for the first time and quantitatively. And so this has potential for human imaging, but because if you can take something that's already approved, it may take only a few years to get to patients as opposed to a completely new approval process, which will take 10 to 15 years to get into patients. We've developed ways of imaging metabolism in animal models, and we're now building instrumentation in our laboratory that we'll use to image first normal human subjects, but then eventually to put in the ophthalmology clinic and look at glaucoma patients. Now I'd like to talk a little bit about Alf Dubra's work. So what Alf Dubra does is he develops high-resolution adaptive optics methods for looking at the back of the eye. And the way they work is he corrects for aberrations or irregularities in the lens in the front of the eye to make sharper pictures of the retina and optic nerve at the back. And technology is used in like the Hubble telescope to take pictures of stars. Here you see these are not stars, these are individual photoreceptors, cone and rod photoreceptors at the back of the eye using the same technique. And Alf is also looking at capillary imaging, imaging of blood vessels to follow some of Andy's results and to look at the blood vessels in particular layers. So here's an example of angiography using conventional methods and also using some of Alf's high-resolution imaging methods where he's getting at really the fine vasculature in the eye that might prove an early readout of some of those early changes in disease. And so the next steps for the team are really to optimize and tune the imaging devices for humans, We're still continuing to use basic biology as well as the clinical expertise from Jeff Goldberg to guide the engineering approach. We are doing studies of healthy eyes for some of the techniques and moving into this direction for others, and we will soon do validation of glaucoma suspects and patients. And I should mention that at Stanford, they're also commencing studies for some therapeutics aimed at protecting neurons and enhancing neurons in glaucoma. And so some of these imaging techniques or new ways of taking pictures of the eye may help us to assess some of those new therapeutic approaches, but will also help us to validate the biomarkers. The real hope here, and we're very enthusiastic about some of these ideas, is that one of these biomarkers is going to enable us to measure a dysfunction in glaucoma before it's too late. So to measure dysfunction at the earliest stage before vision is lost so that we can treat early in disease. And so this is the picture where we have a visual field defect, optic nerve, and nerve fiber layer all change late in the disease. We are hoping and are highly optimistic that some of our new ideas will lead us to a biomarker that changes early in disease and allow us to both detect, treat earlier, but also to develop new treatments. So I'd like to thank you all for your support.